welcome to the second lecture of characteristic of plant growth and development. So, if we recall in the first lecture what we have discussed, we discussed the primary growth and development, secondary growth and development, lateral growth and development, the axis along which the growth and development occurs, the points or the region of the plants where the meristematic activities are positioned, and how they are maintained and coordinated during the process of plant growth and development. So, now we will continue this lecture of characteristic of plant growth and development, and another very important characteristic of plant development is plasticities. It is extremely important for the plants, considering the fact that plants are sessile in nature. They cannot move from one place to another place, which means that they have to face all the environmental disturbance or all the environmental condition just by standing at one place. And to cope with this change in the environment, they have adopted a very nice mechanism and which is plastic in nature. So, the plasticity is basically an adaptability to prevailing environmental condition. I will take couple of example for it, and you will appreciate how developmental plasticity is adopted or it is acquired by the plants. So, if you take this example, this is couch grass. So, the couch grass basically its shoot grows below the ground as well as above the ground. And what happens when the shoots are growing underground? Underground means it has a very different environmental condition than the above ground. Underground, the light intensity is less, oxygen availability, environmental other factors which are very different than the above ground. But what you can see when this grass is growing or when the shoot is growing underground, it has a developmental program which is giving rise to long internode and leaf like structure which is called scale leaf, and more important that at every node. So, here is the things at node you can see lot of advantageous root development. So, this is important thing. So, there is a developmental program, which is going on here already, but once this growing shoots reaches the ground, once it is coming outside the ground, it switches its developmental program. There is a change. The first thing what happens that now it start making short internode. So, short internode. So, the internode length is reduced. To do that, it has to change its the basic program of developmental biology. And what happens here? Another very important thing that now there is a formation of true leaf blade like structure. And the most important thing what is happening that this shoot at the node, it is no longer making the roots. So, what it has to do developmentally? First thing what it has done, it has changed its program for making long internode to the short internode. It has made the changes in their program of the leaf development. Now, the leaf is different types of leaf, and more important thing, when it was underground, it was having active developmental program for advantageous root development, but once it is coming above ground, it has to shut down the program of advantageous root development. This is another case of plasticity, developmental plasticity. This, if you look, water crowfoot, which is a kind of aquatic plants, and it grows in a way that a portion of these plants are submerged in the water, then a portion which is at the surface or at the interface of water and air, and then a certain part of this plant is aerial, which is outside. And that is why this plant makes three types of leaf. The leaves which are submerged, they have a very different morphology, they have a very different structure, they have a different function. If you see these which are basically at the surface of air and water or at the interface of air and water, its morphology is very different than this one, and the aerial leaves, they are totally different. So, this is one example where the program, developmental programs are being changed depending on the environment where this developmental program is happening, what is the surrounding environment for this development to happen. Next characteristic feature of plant development is how cell fate is determined. So, if you remember previous class where we have discussed that the first step of organogenesis or differentiation is cell fate specification or identity of a cell or specifying identity. How this is possible? In animals, this is widely known that the cell lineage is playing a very important role, which means that the ancestors of the cell which is giving rise to the daughter cells, they are also passing the information for its identity. But in case of plant, you will realize that there are some mechanism of lineage dependent development, but which is very restricted, and the major developmental fate is defined by position dependent mechanism, which means that the cell fate is determined by mostly by the neighboring cells who is next to the cells, not entirely by the ancestors. This is important because in case of plants, the cell migration is not occurring. During animal development, cell migration plays a very important role in the development, whereas in case of plant, due to presence of a very rigid cell wall, very tightly glued cell wall, the plant lack the cell migration, which means that the neighboring cell in case of plant development is going to play a very crucial and very important role. So, if you look at the information which is helping for a cell to take its proper fate, it can be either cell intrinsic or cell extrinsic. Cell intrinsic means is it lineage dependent or position dependent. If you look this schematic diagram, if it is a lineage based mechanism, then what happens? If consider this is cell A, this is cell B, they are maybe next to each other, but cell A will already have its own information for specification. B will have another information for the specification, and the cells which are being generated from the A, they will carry this information from the ancestor. So, these descendant cells or the daughter cells, they are going to take the identity or the fate as instructed by their parent cell. But if this is position based mechanism, then the parent cells, they do not have any information for specification, they just divide and give rise the daughter cells. Now, the daughter cells which are next to each other, they pass the information. So, this cell might get information from this cell and this cell might get information from this cell, and based on that they define or they decide what is my relative position in the body, and that helps them to take the identity. And one way of doing it is the clonal analysis. So, in clonal analysis what we do, we use a heritable visible marker such as pigment mutants. So, for example, if you take this plant or in any part of the plant and if you mutagenize them or you expose them for some kind of mutagen and try to find out a mutant where some cells they lack some kind of pigments, let us assume chlorophyll. So, if you take this leaf and if you irradiate them at early stage, what you see that you might find a cell here which is losing the pigment, and then later on at the mature leaf if you see there is a patch of cells which are losing the things. But if you take this leaf and irradiate at the later stage when the development is quite bit completed, then you see that these patches are relatively smaller in size. So, what this tells? This tells that through the clonal analysis you can actually identify the lineage and the cells which are coming from the lineage. So, there is a strong correlation between lineage as well as the position of it. Okay. Another way of detecting or checking this is chimera analysis. So, chimeras are a kind of plant which might have two or more distinct cell population. So, if you take this example, so let us assume that this is a kind of top view of a plant and it has a kind of sectorial chimeras. So, one small portion or sector of this growing meristem has certain marked cells and others they are unmarked cells. And if plants which are coming from this meristem, if you look them, you can clearly see that some regions of these plants which are coming from these cells, they are the marked one. And these two experiments, using these two experiments, you can actually trace out the lineage of a particular cell. Another kind of chimera could be the periclinal chimera. Here what happens, for example, if you look this one, so it is known that the meristem, the shoot apical meristem, it has basically 3 layers, layer L1, L2, L3, and it is known that epidermis, which is the outermost layer of any organ, is derived from the L1, and L2 is usually giving a cell layer which is beneath the epidermis. So, if you mark these cell layers, the L2 cell layers, and then if you make an anatomy of the mature leaf which is coming from here, what you could see that the cell layers which is beneath the epidermis, they have the marked cell. But apart from that, there are other cells as well which are away from these cells, they have also got the marked cell property. But in case of epidermis, it was observed that both the mechanisms functions. So, for example, the differentiation of epidermis is kind of position dependent as well as the lineage dependent. For example, if you look the mother cell, so the specification of mother cell for the stomata or mother cell for the trichome. So, if you look the epidermis, leaf epidermis, there are 3 types of cells, pavement cells and the cells which are going to make stomata or the cells which are going to make trichome. And what has been seen that from epidermis to mother cell formation, it is mostly contact dependent, position dependent, but the mother cell which is responsible for the stomata, it is lineage dependent. Once it is the cell fate is determined, it will follow the lineage and it will make the stomata, whereas the trichome development is basically adopting position dependent mechanism. Another very important technique which can be used to actually distinguish whether cell fate is determined by lineage or by position is laser ablation. So, laser ablation is a technique through which you can very specifically targeted way you can kill some cell and then you look what happens to that cells. So, first example if you take, I am going to kill this, or if you kill the cell which is called quiescent cell center. So, this is a typical apical root apical meristem. In root apical meristem, these cells are very important cells. Why? Because they pass the information, they are very important to maintain the stem cell niche in the root apical meristem. So, all the cells which are directly in contact with QC, they have meristematic capability. And what happens if you kill this QC or if you laser ablate this QC? What was observed that, so if you look here, this is procambium cells. What happens if you kill this QC through the laser ablation? The some cells of the procambium, they comes at the place of QC and then they make new QC. What it means? It means that the fate or identity of procambium is now changed to the QC identity. And if this happens here, the cells which are in this region, they originally they were also procambium cells, they change their identity and then they make new columella cells which is typically present below the QC. So, if you look here, so there is a change of the identity. This suggests or this tells that the information is not lineage dependent. The new fate determination or new QC or new columella, they are taking a new fate depending on their position, not what was their initial programming or what was their information inside it. Similarly, another very important tissue, if you look here in the root tip here, this is QC. In QC, these are the cells which are called initial cells. These are the cells which are meristematic cells, and in case of root there are different cell layers. So, cell layers are like epidermis, cortex, endodermis, pericycle and vascular cells. And what is important here, that each cell, each initial cell, they give rise to one layer of the cells. Particularly here, if you look this initial cell, it will give like this cell layer. But there is one cell which is here, if you look this cell, this is initial cell because it is in direct contact with the QC, and this cell actually eventually give rise two layers. So, the layer of cortex and layer of endodermis, they are originated from a single initial, and this initial is called cortex endodermis initial. So, what happens, this initial first it divides and then it makes a kind of periclinal division, and this periclinal division gives rise to two cells, parallel cells, and one cells then it continues making endodermis, another cells it start making cortex. But what is important here, if you laser ablate this COI, what you see, what we see that the cells which are in the pericycle, this is the inner pericycle layer, the cells they basically are getting pushed in and they are taking the place of CEI. And what happens when they take this place, they undergo the process of a periclinal cell division, and the cells which are outside, they now switch their identity and they become cortex endodermis initial, whereas the cell which is inside, they retain its capability or its identity as pericycle cells. So, both of these experiments suggest that the new cell fate is not inherited or it is not coming from the mother cell, but it is getting acquired depending on the place of the cell where this new cell is positioned. If position is very important in case of plant growth and development, what are these positional signal? How a particular cell realize or how a particular cell acquire a particular identity, how it gets the information from the neighboring cells. And there are certain mechanisms which has been identified. The first mechanism is morphogen. Morphogen are the signaling molecules which basically is responsible for making a kind of, for providing a cell fate or providing identity, and one of such molecule, very important molecule, is auxin. Auxin is a plant hormone. The auxin distribution is very important. It is being transported in the polar manner and there is a formation of gradient. So, auxin distribution, biosynthesis, storage, distribution, everything is important. And the distribution of auxins, some places it makes gradient, which means that the some cells they always get high amount of auxin, some cells they get low amount of auxin. Not only presence or absence of auxin, but the critical amount of auxin is very, very important for switching on a certain developmental program. And another thing which is important with respect to auxin is its concentration. So, some developmental programs are switched by auxin once auxin reaches a particular amount of concentration, which we called auxin maxima. So, if you see here, if you look this picture, so this picture has luciferase marker, but this luciferase marker is driven under DR5 promoter. DR5 promoter is a promoter which has auxin response element, which means that it is responsive to auxin hormone. So, wherever auxin response is there, this promoter will light up. And what you see, you can clearly see a correlation that different regions of the developing primary roots, they have a very high amount of signals, as compared to other regions, and this is called auxin maxima. And another important thing, if you make a correlation, you see that these lateral roots are coming from this region wherever there is high level of auxin or wherever there is auxin maxima. So, this tells that somehow the auxin maxima is correlated with the lateral root development, which means that it might be responsible to initiate lateral root development or the program which is specific for the lateral root development. How it is doing? It might be activating some set of genetic network. Where it is activating? It might be activating in some of the cells which is competent of giving lateral root primordial. So, one mechanism which is through morphism. Another mechanism why a particular cell acquires a particular fate is typical receptor mediated signaling. This is example we will discuss in detail when we will see shoot apical meristem maintenance or root apical meristem maintenance, but here to say that there are some receptors in one cell which is clavata 1 and 2 here, and there are some signaling molecules which is secreted by the neighboring cells, and these neighboring cells they secrete some kind of signaling molecule which is received by the receptors, and then these receptors they initiate a kind of genetic developmental program which is important. So, the second mechanism of acquiring a special fate, position dependent special fate, is through this mechanism. Third and very important mechanism in case of plant is molecular trafficking. So, as I said that the plant cell wall are very rigid, it is very tightly glued, plant cannot move from one place to another place, their position is fixed during the development. Then how they communicate, and to break the barrier of communication, plant has developed a wonderful way of cell to cell communication which is called symplastic cell to cell communication, and this can help in the short as well as long distance trafficking. Trafficking means there are lot of molecules, signaling molecules in forms of protein, in forms of messenger RNA, in forms of microRNA, they have been identified and they have been shown that they are being generated in one place and they are moving to the another place. The movement can be short distance just next to the neighboring cell, or even they can move through the transport system to a very long distance. So, how this happens? If you take this example, this is a protein which is called short root protein. It is a transcription factor, very important transcription factor, and if you look its expression pattern, so here is the transcriptional fusion construct for short root, here is the translational fusion construct. So, what do we mean by transcriptional fusion construct? So, when you take promoter of short root and put GFP downstream it, so this is GFP and this is short root promoter. So, what happens? Wherever this promoter is active, GFP will be expressed and you can see the signal. But in translational fusion, what you are doing? You are taking short root promoter, you are taking short root gene and you are just removing the stop codon and translationally fusing with GFP. So, this is you are basically fused short root protein with the GFP. So, here what you can do, the first thing is that the promoter will ensure the domain of expression and this protein, the GFP will ensure the localization of the short protein. And if you compare these two things, what happens in the transcriptional fusion, where just the transcriptional domain where the gene is getting transcribed, you can see this is the root tip. So, this is endodermis, this layer is endodermis and this entire layer is the vascular tissues, and what you can see, signal is only in the vascular tissues, which means that short root promoter is actively transcribing only in the vascular tissues, proteins are being made only in the vascular tissue. But when you track the protein, short root protein, what happens that the proteins are present in the vascular tissue, but they are also present in the endodermis. This is very important. So, from where this protein is coming, and another interesting thing you can see here, that in endodermis protein is localized to the nucleus. So, from where this protein is coming, which means that one thing is clear from here, that there is a protein mobility. Short root protein which is getting synthesized in the vascular tissue, it is moving to the endodermis, because in endodermis transcription is not happening. So, gene is not getting expressed in the endodermis, the endogenous gene is not getting expressed, but protein is present. And then we know that this is happening through a very special structure in case of plant, which is plasmodesmata. Plasmodesmata are the nanopores which basically is between two cells, and they are providing a path, a symplastic path through which molecules can move from one cell to another cell. But there are a lot of questions. Is the movement free? No. The movement through the plasmodesmata is highly regulated. So, only those molecules can move through the plasmodesmata which are supposed to move. Those molecules which are not supposed to move, they cannot move, because if everything moves from this cell to this cell and everything moves from this cell to this cell, then both the cell will lose their own specific identity. So, both the cells, they are ensuring their specific identity, but at the same time they are allowing certain molecules to move from each other, from one cell to another cell. And this is a very unique and very important feature which plant cells has acquired during the process of development, and playing very important role in plant and growth and development. We will cover this in detail in the later classes. But if you look this picture, is short root protein moving through the plasmodesmata, and how it has been tested. So, there is a mechanism, or there is a program through which what you can do, you can actually block the plasmodesmata between endodermis and the vascular tissues. So, as in the previous slide I showed you that these proteins, or the short root proteins, are being made, are being transcribed and translated in the vascular tissues, and then protein is moving to the endodermis. If somehow we block the plasmodesmata which is present in the cell wall between endodermis and the vascular tissue, we should block the movement. And this is what you can see here. When we block this plasmodesmata, you can clearly see that the signal, short root protein signals, are disappearing from the endodermis. Look here, this is completely disappeared after 24 hours of the blocking plasmodesmata. So, this clearly tell that the critical or very important transcription factor, short root protein, is basically getting moved or getting trafficked or getting transferred from one cell to another cell, specifically through plasmodesmata. Another important characteristic feature of plant growth and development is totipotency nature of the plants. What it means? Every cell in the plants, in principle, they have a capability that they can undergo the process of cell division, cell differentiation, dedifferentiation, anything, and they can make a kind of plant. And imagine why this is so important. Again, I would say that plants are sessile, they cannot move. So, they have to have a mechanism, a robust mechanism, that under adverse condition, if everything they lose, even they retain some kind of living cells, they should revert back and they should propagate back to that one. So, this slide, if you look here, there are some natural mechanism of regeneration, and this regeneration mechanism, there is a genetic program, there is a regulatory program for this process. Some of them we will discuss here. First thing, if you look here and here. So, if somehow the root tips or shoot tips are damaged in a plant, or if you cut them, what happens? You know that the apical meristems are positioned in the shoot tips and the root tips. If they are lost, so there is a mechanism, plant has acquired a mechanism that they can actually reconstruct the meristem. This is because of their regeneration capability. Another important thing, if there is a damage, if there is a tissue damage, again plant has a mechanism through which they can repair the damage. This is again getting activated through the process of regeneration. One thing which I discussed in previous class was the apical dominance. So, what I said that when plant is primarily growing, then there are axillary meristem. Axillary meristems are specified, but their growth is inhibited because plant first want to ensure a certain amount of growth, particularly they want to transit to the reproductive phase, they want to ensure first successful reproduction, and then the signal is being transmitted and lot of axillary branches they started growing. But what happens if you cut the plant from here? They immediately sense it, and since this is cut in the differentiated region, if you cut in the meristematic region they can reconstitute the meristem, but if you cut the region which is fully differentiated, it is difficult for them to fully reconstitute the meristem. But what they does, they immediately activates its axillary meristem and they started making the branches. So, axillary shoot outgrowth is now getting activated. Another very important thing is de novo organogenesis. So, this is a property of plant cell. If you take any part of the plant, which typically we are using in the process of tissue culture, which we called explant, and if you take this plant, there is a genetic program through which you can activate a proper genetic program and you can generate a full plants or full plantlets. For example, you can take shoot and you can induce the root, you can take shoot and you can induce the shoot. Similarly, you can take shoot and you can induce both shoot and root. You can take root and you can induce the shoot. So, what it tells that? It tells that there are the genetic program. If you give a proper signal, if you give a proper information or proper hormonal combination, you can eventually activate a full and proper developmental program for the shoot development as well as root development. Another important developmental program which occurs is the lateral root formation. So, as I said, when plant is growing in the apical and basal axis, it is growing by using its apex or shoot apical meristem and root apical meristem. But once these tissues they are entering in the region of differentiation, there is a new meristem which is getting generated somewhere here, and this meristem they acquire a capability and they make lateral root primordia, and then they eventually they make lateral roots coming from here. So, what happens? There is a difference between apex. So, apex, at the apex the meristems are specified as a meristem and they remain as a meristem. So, they are undergoing only the process of cell division, but here what is happening that at fully differentiated cells they are undergoing the process of de-differentiation, through which they are losing their existing identity and they are acquiring new identity. First the meristematic identity and then later they undergo a new round of differentiation program or totally new differentiation program, and now they are making a new organ or new tissue. So, the plant cells they also have this capability that even though if they are differentiated, they can initiate, if proper signal is given, if required, they can undergo the process of de-differentiation and they can undergo the process of redifferentiation. So, if we summarize all the characteristic feature of plant growth and development, so what we have seen that plants are sessile and they are actually very prone to the physical damage, and that is why they have evolved a developmental pattern that are continuous, responsive to the environmental cues and regenerative in nature. These features are attained in large mostly by exchange of the information between the cells, which can be local as well as long range, and the cell fate in the developing region is more dependent on the positional cues as compared to the lineage information. So, we will stop here and we will discuss in next class. Thank you very much.